So, dzień dobry. Dziękuję za zaproszenie. Cieszę się, że tu jestem. So, I try to be on time, and I hope I will be able to answer this very difficult job to define the best therapy for myeloma. And of course, in the field of myeloma is very lucky, although it's a very unlucky disease. We are a family of myeloma experts, and all over the world we are very lucky to be sharing the same language and hoping for the best of the patients. And with this, I would like to describe that what we had earlier is a reference to what we are facing for future. And when you say best, we have to define what is the new development, and so what it adds on top of what we have currently. During the last couple of years, we learned that the old fashion of using one single treatment like PI-based or IMiD-based approach was not enough, and so combining regimens, an IMiD plus a proteasome inhibitor was the best approach. But with the advent of monoclonal antibodies into the field, and the relapse scenario changed, and today we know that we have to modify our regimens, not based on the general approach, but also a kind of a patient-based approach, and this includes two types of criteria. One is for risk definition, and the second one is the patient perspective, the tolerability and the frailty of the patient, which will define the design of the protocol the patient should be receiving. So, in addition to these two main factors, we know that the triplet regimen is a better approach. It increases response rates, and it's now even possible for elderly patients. And the advent of and the introduction of monoclonal antibodies has now changed the treatment scenario for both the elderly and also the young patients at diagnosis. So what was observed in the relapse setting has now become applicable for the newly diagnosed patients. And stem cell transplant, we have stem cell transplant experts here, is always being questioned. And the European Myeloma Network study and also the IFM and DNAbased study was questioning the role of delayed transplant, but eventually it ended up that with use of VCD or VRD as an induction, stem cell transplant is still needed. But will that be the same situation in the near future? We will question this. And there are also concepts for high-risk patients. When it comes to high risk, the role of transplant is stronger, and even double transplant has been shown now in Europe mainly that it is required, and it increases the PFS and of the patients. And nowadays, it's no question that maintenance therapy is as long as possible, at least for 2 years, and as long as the patient tolerates. That this is a new concept. While it was being questioned a couple of years ago, now there's no more any question. Maintenance is one of the mainstays of myeloma treatment. So when you look at up-to-date, in 2019, they describe the risk status based on these FISH parameters. So there are 3 translocations partnering chromosome 14 with 16, 20, and 4. These are the 3 risk factors. And in addition, on top of it, there is deletion 17p and also 1q amplification. But it's not so easy. We can talk about an hour about this topic. So what is the cutoff for 17p? Is deletion important? or is the mutation more important? Is it monoallelic or biallelic deletion? Which one is more important? And very recently, 1q amplification has become another topic of great importance, and number of copies, more than 5 copies, is more important than smaller number of copies. And about risk assessment, based on these, at least these parameters, we now know that there is standard risk or high-risk patients. And it's not only the age now which matters, but the risk status is more important. And as long as the patient is high risk, it's better to force treatment. And up to date, it's the triplet regimen, PI plus IMiD, and to enforce this with stem cell transplant as long as the patient tolerates. If not, the treatment should go up to 8 or to 12 cycles, and then the maintenance starts with, here in this scenario, it's bortezomib. So for high-risk patients, maintenance with PI regimen is the main core of the treatment. For those who are not high risk, which is usually the majority of the myeloma patients, in that case, the stem cell transplant option is an important one. And with this, we either, the usual trend is to give 4 cycles of VRD regimen and perform a transplant. And here the discussion is to do immediate transplant or delayed transplant. But in that situation where transplant is delayed, then we have to continue treatment with maintenance. And here the maintenance is not with bortezomib, but instead with lenalidomide. So these are the main agreed evidence-based situations in 2019, but things are changing very fast. And we know that if one patient has more than one abnormality, so these FISH abnormalities that I described, is if it appears with more than one, so double hit, like double hit lymphoma, there is double hit or triple hit myeloma, and that's an ultra high risk, and that refers to a very bad prognosis, and it's seen about 10 to 15% of myeloma cases. And how many courses of induction should be given? And usually in the up-to-date you have seen 4 cycles, but the Spanish people were the pioneers to increase the induction regimen cycles, and next week in Boston this is again going to be presented comparing different trials. So it looks like that 6 cycles may be more optimal before transplant is done, as long as the patient is responding. And I'm going to be showing you in a minute monoclonal antibody combinations, but monoclonal antibodies certainly are very beneficial, but should all patients be receiving monoclonal antibody at diagnosis and during the early phases of treatment? And what about the minimal residual disease story, which is another hot topic which has been now one of the endpoints of clinical trials, and now also has been approved by insurance companies as a surrogate marker, and sometimes is a topic, should that be discussed for end of treatment target. So I will now concentrate on 2 parts of myeloma treatment. Patients who are not eligible for transplant is the first part, and the next will be the transplant-eligible patients. Tradition-wise, VMP in Europe, the VISTA protocol, was a major breakthrough which made bortezomib the backbone of induction regimen, or the main treatment modality for all patients, not only for transplant eligible, but also ineligible patients. And this was followed by lenalidomide, dexamethasone, and these 2 studies were then questioned with combinations of quadruplets or sequential versus alternate regimens combining VMP-RD approach in different modalities, 9 VMP courses followed by 9 RD courses, or sequentially. And all these ended up, you can see the comparisons. The median PFS started from 20 months to 32 months. With the increase in duration and with longer treatment, the response is more durable and higher. And the biggest achievement in the PI and IMiD era is the introduction of VRD. So it's the VRD light regimen, which was published by 2 groups. And this has increased the PFS value to 43 months, and with an overall survival of 75 months, which was unimaginable in the earlier times of myeloma treatment. And this is the publication from the US group led by O'Donnell and senior author Nopuraja. And this light regimen gives the beauty of being tolerable by elderly patients as well, not only for younger ones. And this led to the approval of bortezomib and lenalidomide in this year as an approved regimen combination for transplant ineligible patients. So for the future, what we have achieved so far, is this enough? Of course, nothing is enough. I mean, the worst enemy of the best is the even better one. So we have the monoclonal antibodies, which made a breakthrough in relapsed patients. And here is the example, which also led to approval in first in the United States and then later in Europe and also in other countries, including Japan. So VMP, the VISTA regimen, has been improved with the introduction of daratumumab, anti-CD38 monoclonal antibody. And the control arm, you see, is continuously decreasing. Why? Because after a while, you have to stop VMP. Elderly patients do not tolerate bortezomib for a long time. You have to stop at some point. But continuing with daratumumab single agent, that was enough to improve the response. And with a hazard ratio of such a big difference, there was, which led to an approval. So although melphalan is not very popular in the United States, it's still controversial in Europe, where melphalan has always been popular, very easy to be applicable. But with bortezomib, this was the case. And so is there another one, another combination of monoclonal antibody? Yes, there is, and this has been also the final story of the MYELOMA study, which was published this year in May. Again, a very good improvement. RD, you remember, is almost comparable to VMP, but it gives the durability and also long-term usability and tolerability. And so adding a monoclonal antibody on a more continuous treatment approach was able to improve responses, almost like a transplant outcome, and again led to approval by FDA. And the approval in Europe is not obtained yet, but it's under evaluation. So here you can compare different regimens. The blue ones are the PI-based ones, and the colored orange and yellow are the ones on the IMiD-based regimens. And the control arms are also comparable here. The DARA-VMP ALSA-1 study, which we participated in as well, and the MYELOMA study, DRD, the median PFS values have not been reached. So here currently the scale is up to 55 months, but it will be maybe even better in future when the later long-term results are reported. And so for today, for transplant ineligible patients, there are 3 standards of care depending on the access to drugs in the country and also the insurance companies' payment status. So VRD, which is in the middle, you can see that younger patients Although they are not eligible for transplant, they could be younger and they could be elderly. And for those who are very elderly, the difference between RD versus VRD is still important. But the difference and the absolute values are even better for younger patients. When you compare DARA-VMP versus DARA-RD in the same scale, you see that there is a bigger advantage in the MAIA study. DARA-RD is the best so far. Why? Because it's more longer treatment. So for myeloma treatment, you have to continue treatment, and the more tolerable, the less toxic the treatment, and more efficacy, then we see the outcome as a big success. And MRD values also reflect the depth of response, and here, based on these, the algorithm in 2019 is a little different than what is seen in the up-to-date so far. Up-to-date has to be revised, I guess, and we have to add the fourth combination, which is the monoclonal antibody VMP or RD, as another alternative for the standard treatment, and we have to acknowledge that standard versus high risk must be always determined at diagnosis. And for those who are not transplant eligible, the new questions are, is there anything to improve VRD? If you visit Mayo Smart site, you will see that KRD is a standard for high-risk patients, but in the up-to-date version, we do not see that yet. So second-generation PIs, such as carfilzomib, is now being questioned here, and triplets and quadruplets are also being compared. Adding monoclonal antibodies is the same style is also present for these kind of patients. And again, the question of induction cycles is questioned in some of these. And when there is a transplant question, there are things that can be improved with transplant. It's not only the number of transplants, but also the conditioning regimen has been questioned. But again, this is another big topic, so I just briefly mentioned that combining two alkylating agents or adding PIs such as bortezomib or carfilzomib or lenalidomide or bendamustine to the conditioning regimen, these have been all questioned in prospective manner and also in retrospective manner. And at ASH, we are going to present an EBMT study looking at the value of adding bortezomib to melphalan in more than 300 patients in the EBMT registry. So the UK people are one of the leaders in this field. In this study, which is myeloma 11, they have accrued more than 3,000 patients in a very short time period in more than 100 centers in the United Kingdom. And they are comparing two triplets versus a quadruplet, and based on the response which is achieved with CTD, cyclophosphamide, thalidomide, dexamethasone versus cyclophosphamide, lenalidomide, dexamethasone. If the response is PR, then in that case they randomize to receive bortezomib with cyclophosphamide or not. But if the response is less than that, then they continue with VCD regimen. And KCRD is the other one. And we have seen the reports of this study at EHA 2 years ago, and there it was shown that the quadruplet regimen was given less. So 4 versus 5 cycles. I think this is a big message. If you increase the number of drugs within a combination, you need less number of cycles. And the response achieved with the quadruplet is much better. So the winner in the PFS value is for the quadruplet. Another big group, GMEMA, the Italian people, have done another study which rather questions the value of transplant in a setting where there are 3 arms. One is similar to, in a way, similar to the British study, where one arm includes, all arms include carfilzomib, but IMiD-PI combination are the two parts of the study. The aim is to continue with only transplant 12 cycles, whereas the third arm is without any transplant. So transplant versus with or without transplant is the main question here. And finally, what the results are. The results are very impressive, and it shows that KCD regimen as an induction is not as strong as KRD. That's for sure. And if you do transplant with KRD, you improve the responses compared to KCD. But if you give continuously KRD, the results that you give are almost as similar to without doing a transplant. So is this VGPR or CR numbers valid when you look at MRD? I guess so. I think the difference of 58% versus 54% of MRD comparing with or without transplant are almost similar. But we have to see the long-term effects. In the IFM Dana-Farber study, it was shown that with transplant MRD was deeper. So we need to have longer follow-up of this study, which is still ongoing. So the introduction of daratumumab this year was another breakthrough, and the collaboration between the HOVON and IFM results were presented at two major congresses and very recently published. Cassiope study was comparing VTD, so the old-fashioned IMiD thalidomide combined with bortezomib plus minus monoclonal antibody against daratumumab. That the results are showing that addition of daratumumab to VTD was very effective and with very good PFS values, hazard values. Of course, the overall survival impact is too early to see, and this has been very recently published in Lancet. And again, this has led to another approval. So is VDT as effective as VRD, and should this combination of daratumumab with a PI-IMiD-based regimen be VTD is the partner or VRD is the partner? So US people have started the Griffin study, which was a phase 2 study, which was finished, and earlier this year, there was an announcement in the newspapers, but I think there is a typo error here in the message. The p-value should be bigger, because the difference is quite big. So the earlier results were published as positive, and the phase 3 study, the comparison study, is ongoing in both US as a Griffin study, and in Europe there are two studies, both for the younger and elderly patients, Perseus and Cepheus. We are participating in both of these studies. Perseus is the transplant eligible, Cepheus is for the elderly, DRD, VRD versus VRD regimen. And one is the European Myeloma Network study, the Perseus for younger patients. The other one is a pharma-sponsored study. And certainly monoclonal antibody, anti-CD38, daratumumab is not the only drug. Isatuximab, a Sanofi product, is another one. And IMRO study was very quick. We also participated in this study for the elderly, but there's also another version for transplant-eligible patients, which is, the study is done now in Germany, and so we will soon be seeing results of VRD combinations with monoclonal antibodies, hopefully next year, of the combination of VTD is inferior to VRD or not. And again, another question, we now know that KRD is a good regimen, so adding DARA on top of KRD, will that also improve? So there's another study which questions that. And in France, the frontline proteasome inhibitor has now moved to ixazomib, the oral formulation of again produced by Millennium, the same company. So, and another antibody, elotuzumab, but it's another target, anti-CS1, is also being questioned. So for future, to improve the induction regimens, we will have the results from the monoclonal antibodies, and hopefully we will be able to see that that maybe transplant will no longer be needed. But still, I think transplant is needed, mainly for patients who are really very high risk, such as those with extramedullary disease. And now we have data evidence that for high-risk patients, EMN02 study, and there's an EBMT study for extramedullary disease that shows that transplant is really needed at least once. And we also know that transplant is a good salvage treatment based on the response duration after the first one. So, and for maintenance, I will be very quick. We know that lenalidomide is a very good drug for maintenance, and now there are going to be 2 studies which are going to address the role of ixazomib, and we will hear those. But from the myeloma 11, we now know that the role of maintenance is valid not only for transplant eligible, but for also non-eligible patients. And when we look at those who are in the eligible group, the overall survival benefit is also present. And across all risk groups, the difference is visible. So for all patients, not only high risk, but low risk, those patients should receive lenalidomide. But you remember in the algorithm for high risk, it's usually a proteasome inhibitor which is selected. So this has not been tested yet, but it's based on results from individual studies. So lenalidomide is a good drug for maintenance, but proteasome inhibitors are also. So for finally, as a before closure, I want to make the platform ready for gene-based analysis, looking at individual differences patients carry, not based on their FISH criteria alone, but also on their gene expression profiling. Why? Because gene expression profiling has been shown by mainly the Arkansas group, and also the European HOAN group has been very instrumental in looking at carefully the data. And here in this study, which was published like 2 years ago, they were able to kind of predict the lenalidomide sensitivity, but none of these genes are specific for a pathway. It's more like a general view, and they were able to test this in different patient groups. But this is not a prospective study, so it has not been tested prospectively. And the HOAN study was able to again do a similar approach for lenalidomide and bortezomib, and they were able to clear and show some differences in terms of predicting sensitivity. Finally, this is the MMRF study, which is a MyDrug study, which is based on the targetable gene mutations which can be shown. I know that in Europe, in many places, next generation sequencing is becoming widely applicable, and in those patients who have mutations which are targetable with the drugs which are used mainly for solid tumors or others, these can be combined and integrated into a myeloma study. So this is a newly diagnosed, but maybe has more role in the relapse setting. So with this, I would like to conclude, and I have to acknowledge the importance of collaboration. Myeloma or plasma cell disorders are rare diseases, and we need to be sharing the same goals and collaborating to be able to achieve faster responses. And I'm really grateful for being able to be here. Thank you.